Hello, everyone. My name is Sam Morningstar. I'm a member of the Golan Evans Lab at the Center for Regenerative Medicine at Boston University. I'd like to present my work today, which is titled The Investigation of VEGF mRNA-LNP to Reduce the Liver Disease Burden Through Generation of Healthy Hepatocytes from Biliary Duct Cells. Now, we're conducting this research in the context of alpha-1 antitrypsin deficiency. So alpha-1 antitrypsin deficiency is a genetic disorder which causes the misfolding of the protein AAT in the liver into the Z conformation. This leads to it being unable to be secreted by the hepatocytes, which eventually leads to hepatocyte death, fibrosis, and cirrhosis. Now, alpha-1 is a genetic disease with no cure currently aside from liver transplant, but due to the scarcity of available livers, patients often die while waiting for a transplant. Thus, therapeutic alternatives to treat this disease are essential. And we use the NSG-PiZ mouse model to recapitulate this liver disease phenotype in mice. On the left here, you'll see a human patient with alpha-1 antitrypsin deficiency with a PAS-D staining, has these magenta inclusions, which marks the misfolded Z-AAT in these liver cells. And if you look on the right, this is the NSG-PiZ mouse model. We also see in a PAS-D staining these magenta inclusions. Thus, this is a good model to study the liver disease phenotype in mice. To treat this disease, we're using our mRNA-LNP technology. Now, mRNA-LNPs have been previously validated by the lab as a method to effectively and transiently express proteins of interest in the liver. In our paper, upon intravenous injection of EGFP mRNA-LNP, we could transfect 100% of hepatocytes with this GFP, as well as 70% of endothelial cells and 40% of Kupffer cells. And importantly, upon transfection, when we looked through IVUS of luciferin, we saw that around 72 hours, the protein of interest began to fade from the system and leave. So this is an effective method to transiently express these proteins of interest. So, also previously published in the lab, we've shown that through injections of VEGF mRNA-LNP, we can stimulate this biliary epithelial cell to hepatocyte conversion in both chronic and acute liver disease models, and that injections with these VEGF mRNA-LNP showed decreased levels of steatosis and fibrosis. Now, this see in this lineage tracing model with a mouse with high-fat diet, we showed a marked increase in the amount of Beck-derived hepatocytes upon injection of VEGF mRNA-LNP. And interestingly, in both the CDE chronic disease model and in an acetaminophen overdose acute disease model, these biliary duct cells show expression of KDR, which is the VEGF receptor. So our hypothesis is that through utilizing this VEGF mRNA-LNP to stimulate the innate Beck-driven liver repair mechanisms, we can create a new potential bridge therapy for patients with alpha-1 antitrypsin deficiency waiting for a liver transplant. So we use this chronic disease mouse, and we use the VEGF mRNA-LNP to generate healthy hepatocytes, which can then extend these patients' lives longer so that they can live long enough to receive an available liver. So in order to study this in this model, we use 3 different transgenic mice. So first we have the NSG-PiZ mouse model, which will recapitulate the liver disease phenotype in mice. And we cross homozygous NSG-PiZ mice with homozygous TD tomato reporter mice. Now, mice that are heterozygous for both of these genes will then be crossed with a homozygous Beck-specific CK19 Cre mouse. So then the offspring of these were genotyped, and only mice that are heterozygous for all 3 of these genes will be used for experimentation. This ends up being around 25% of the mice. So this allows us to do lineage tracing in this mouse model for biliary epithelial cells specifically. So around 6 weeks of age, we'll inject them with tamoxifen to induce this lineage tracing. At 8 weeks, they'll receive AAV-TVG-P21, which will increase senescence levels in hepatocytes to levels that are consistently observed in human patients with alpha-1 antitrypsin deficiency. And then we will treat the mice with either VEGF or control mRNA-LNP and sacrifice at varying times to see the effect on cell conversion and to see if we can have any effect in alleviating the disease burden. So first, before we wanted to conduct this research, we wanted to validate our triple heterozygous mouse model to see if it still had the common traits associated with alpha-1 antitrypsin deficiency. So this leads to our first figure, in which we were comparing a PiZ homozygous mouse to a PiZ heterozygous mouse. We looked first at PAS-D and 2C1 staining, which marks the mutated Z-AAT in the liver. And as we saw in these stainings, the heterozygous mouse does still express the mutated Z-AAT, as shown by the magenta inclusions in the PAS-D staining and the red staining in the 2C1 staining, although it appears that the cells express less mutated Z-AAT per cell. It seems that it's still similar distribution per cell across the liver lobes. We also wanted to look at cell proliferation through EDU staining to see if these heterozygous mice have similar levels of EDU, and we saw similar EDU stainings across our heterozygous and homozygous mouse models. And we also wanted to look at ductular reaction, which is another liver disease phenotype, which generally precedes fibrosis in chronic liver disease, and we see marks of ductular reaction through our CK7 staining, where you have the positive ducts, but also these branching CK7 positive cells, which mark the ductular reactions. So, upon seeing that the heterozygous mouse does have similar liver disease phenotype to the homozygous mouse, this was reassuring and allowing us to further conduct experiments using this heterozygous mouse model, which leads to my figure 2, which is the treatment scheme I showed before, except so we treated the mice with tamoxifen and then P21, and at 10 weeks we gave them either VEGF or control mRNA-LNP, and then we gave them more injections at week 11 and sacrificed at week 12. So what we were happy to see is that upon treatment with VEGF mRNA-LNP, we were able to generate hepatocytes that were tomato positive, thus Beck-derived. This is shown in panel B, which we have this cell with a hepatocyte morphology that also expresses tomato. So it was good to see that we were able to generate these cells from the biliary duct cells. Also, interestingly, we see tomato positive area in the nuclei of some hepatocytes, as well as tomato positive area in the bile canaliculi of some of these hepatocytes. Now, further quantification and classification is needed to be done through co-stainings with CD26, as well as hepatocyte marker HNF4 alpha to confirm the hepatocyte identity. But we were happy to see that in this early data, we can still generate what appears to be healthy hepatocytes from these biliary duct cells. However, further quantification is needed. Now, because we didn't see as much cell conversion as we'd like, we went into extending the lifespan of the mouse longer following the VEGF injections to see if we can increase the amount of clusters of hepatocytes. So this is figure 3, which is the same treatment outline. It's only we extend the mouse's lifespan to week 14 and then sacrifice to see if this has an effect on the amount of tomato positive hepatocytes that we see. So in panel B, we showed what appears to be clusters of cells that are negative for cholangiocyte marker EpCAM but are positive for tomato. This leads us to believe that these cells are Beck-derived, as they're tomato positive and EpCAM negative. They must be Beck-derived hepatocytes because they lose their biliary epithelial cell markers and still express tomato. So this, we can see clusters of what appear to be healthy hepatocytes in panel B. However, interestingly, when we look in panel C, we also see these tomato positive EpCAM negative clusters of cells, only they have these dark circles inside. Now, we believe that these dark circles are likely the misfolded Z-AAT. Because this is a genetic disease, it would make sense that the cells do still have the gene for the misfolded protein. But one thing we're interested in looking into is the maturation of these cells to see how long they live until they begin to express this misfolded protein, and to see if we can further classify this model to see different ways we can generate more healthy cells or prevent the generation of unhealthy hepatocytes. So in conclusion, here we were able to generate what appears to be more clusters of healthy hepatocytes, but when we extend the mouse's lifespan, some of the hepatocytes do begin to express this misfolded protein. However, further classification is needed to be done for this model. So for some conclusions, we saw that the heterozygous PiZ NSG mouse model exhibits similar AATD liver disease phenotype as the homozygous PiZ mouse, such as the accumulation of Z-AAT, cell proliferation, and ductular reaction. However, further quantification needs to be performed here. And our preliminary data suggests that injections of VEGF mRNA-LNP do promote the generation of healthy hepatocytes from biliary duct cells, as well as diseased hepatocytes from biliary duct cells. So for future directions, we want to further characterize this model through immunostainings for liver disease burden, cell proliferation, and ductular reaction. We'd also like to see how increasing the amount of VEGF mRNA-LNP injections, as well as changing the time of sacrifice, can improve the numbers and size of the newly generated hepatocytes. And we'd also like to look into quantifying the amount of liver disease burden in these triple strain heterozygous mice compared to the homozygous PiZ littermates, and further to see if the amount of liver disease has any effect on the amount of cell conversion that we see, to draw a correlation between disease burden and cell conversion in this model of alpha-1 antitrypsin deficiency. So thank you for listening to my talk. I would like to just acknowledge all the members of my lab who've made this work possible, as well as our UPenn and Acutus therapeutic collaborators for providing us with the LNPs that we use to study this. Thank you so much.